Okay, let's start. First thing, a couple of you pointed out a problem in the grade book. Is everybody seeing the same problem? That you're not having, that not all the columns are adding up? They should?
Yeah.
Okay, I'll see what the problem is today. I have to go out of my way to exclude grades from the grade book. So, I have to, if it's a quiz or an exam, I have to actually check, do not include this in the grade book. So I'll see, there was a problem that I got an email about a little bit earlier, but I can't see your grades. There's a dummy student account, but it doesn't really work very well. So I'll get that looked at today. Any other questions? Yes, ma'am.
Were you talking about like everything adding up? Because I don't know about that. I was just talking about the quiz in particular. Because if there's 10 questions, were they all like 1.25?
Okay.
Like that's not talking about.
Right. On some people have been telling me, and I don't know how this happens, but that the grade will be 1.3 instead of 1.25. Are you seeing that?
Well, I'm seeing like
All of them should be 1.25.
yeah, and then there's 10 questions. And
Right.
So it's not. That isn't.
It's not adding up correctly?
Yeah.
Okay.
And right now it says out of 13, but the most
Okay.
you can get is
12.5.
or 11. Yeah.
12.5, right. That's, I hand code each individual question with 1.25, and then for some of you it seems to be rounding up the total. Okay, and probably what I'll do is I'm terrified that I'll reset the grades. I'll probably end up downloading them to an Excel. I can download everything to an Excel spreadsheet, and it should give me the totals that way. Okay. But I will go up there today and ask them about that, because a couple of people have pointed out that if you look at the points, they're 1.25, but it totals and rounds it up, and I don't understand why it does that. It doesn't do it when I look at it, but that doesn't mean anything. Though when you go in as a student, and there's a dummy student that I can use, but I think the dummy student account shows me what I see and not what a student sees. Any other questions? Okay. So we're going to just finish up with this again just a little bit before we start talking about ethylene. And just to reinforce the point that in the root tip, that you can see, if you see a micrograph, you can see these amyoplasts. And if you remember from week 1 or whenever, the amyoplasts are plasts that store starch. Basically, are always going to be collecting at the bottom point, at the lowest point of the cell, wherever the gravity. So they're going to be pointing toward the center of the earth. And then this then causes a very complicated reaction series that ends up with distributing auxins. And in this case, there's a hyperconcentration of auxins on the lower side, so that then the root grows down. So even if there's a block here, so let's say this root is growing along a rock, and you may have seen pictures of this where, one thing that they do when they look at root, the way roots are laid out, they'll cut down in the soil. And if the rock even has any little indentations in it, you can see the root will go down a little bit, and it'll actually go up a little bit as it grows, and it'll go down a bit, and then finally gets to the edge of the rock, and then it grows down. So in this case, you're getting a hyperconcentration, and if we go back up here, and in the case of the chute tips, we're having an oxygen concentration cause the bending toward the light, okay? So it's a different concentration, and these sort of experiments you can do. And these were done. This is why oxygens were worked on so early. It was because it was easy to do these experiments. You could cut the top off of an oat seedling and see what it does. Okay, questions? Okay. Today we're going to pick up then on ethylene. And ethylene's a very interesting hormone. And because first, of course, it's a gas, it's very commercially important. For years, supermarkets, tomato producers, a lot of fruit and vegetable producers have tried to manipulate ethylene by either eliminating it or adding ethylene. And we're going to start off with a little bit of the background. But you'll need to understand a bit about the pathway. It's a very short pathway. And then later on we'll talk about how ethylene and auxin interact to produce true abscission, which is the leaf drop. We'll spend a bit of time talking about how places, even supermarkets, can manipulate ethylene. And we'll spend a little bit of time on the genes that are involved in ethylene action. Now, what's the purpose of this? Did anybody have these? Keep the air moving, yeah, they don't work at all. The notion here is that ethylene is a gas, and so you have your bananas hanging there, and these bananas produce ethylene. And what is weird about ethylene is autocatalytic. What does autocatalytic mean? Autocatalytic. Ethylene causes ethylene to be produced. So we have a backwards hormone. We have a hormone that once a little ethylene starts being produced, it causes a tremendous amount of ethylene to be produced. And the classic, it goes back to when I was an undergraduate, is one bad apple spoils the barrel. Because, fortunately, way before my time, apples were harvested and stored in barrels, usually in sawdust. So you'd have a little bit of sawdust, put the apples in there, put some sawdust in, put the apples in. You keep doing that over and over again. But if one of these apples was bruised to the point of where one thing that happens when cell tissue, if you hit the apple hard enough, you're going to damage the cell tissue, which causes ethylene to be produced. Almost invariably, if you have any plant tissue that is damaged, it's going to produce ethylene. So the notion would be then that ethylene would not only cause that apple to start going past being ripe, and the purpose of an apple is not to be for us to eat it. The purpose of an apple is to rot and get the seeds dispersed. So then that apple would start producing ethylene. It would start breaking down, which releases more ethylene. And what the sawdust actually was used sort of as a cushion, but also it actually absorbs a little bit of ethylene. Then slowly all the apples would be ruined. This thing does not work. Well, if you were able to get bananas that had not been zapped with ethylene, that had been picked green, which is hard to come by, then it might give you a little bit of time. But once one of the bananas starts turning yellow and starts producing ethylene, unless you had a fan blowing really, really fast on here, this doesn't really do you much good. Now, if I'm lucky enough to go to this, And you can see part of it here. This is something that I showed you the first of the semester. And these are green bags. And you can still get these in the store. And any of you have these, you can actually store produce in them. And then from this, we made this. And I don't know why this is off-centered a little bit, but this is linked. This is on the website. So this is an experiment a student did several years ago where this is a green bag, and these are not. This is the green bag. This is just a banana laying out. And here's a banana in just one of the bags that you buy, that you get at the supermarket, one of those incredibly thin polyethylene bags. And you can see that actually the banana in that green bag doesn't ripen as fast. So this green bag actually absorbs ethylene. And if you go to the website, they tell you something ridiculous like it's some chemical mined from a cave in Japan or something like that. In all probability, it's a chemical called potassium permanganate or whatever. And so they do work, and you can ooh, there it blows up now. Now you're seeing a lot of respiration. You're seeing a lot of buildup of CO2, and here we got a lot of nice bacteria collecting there. So these things actually do work. Ethylene does a lot. And ethylene, there's seeds that will germinate that are dormant unless you treat them with ethylene. And they have a large role in anything that has to do with maturity to an endpoint in plants. So development of a flower, development of a fruit, ethylenes can be involved. So don't worry about these individual things, just the fact that ethylene's involved in a lot. Ethylene is an incredibly small molecule. It's a gas, and it's hard not to find ethylene in tissue if you have a sensitive enough instrument to measure it. The instruments to measure ethylene, this was probably the first commercial instrument that had to do with hormones, was of course ripening, slowing ripening. Want to be able to measure ethylene, so one of the first laboratory grade instruments that was modified for use in the field. And by the field I mean in a packing plant, to be used by someone that wasn't trained, was to measure ethylene. Senescence, ripening, we see that. C2H4 is what ethylene is. It is very important. The classic, of course, is tomatoes. Another very important thing is ethylene causes ethylene to be produced. It's autocatalytic. And if you think about it, when one fruit gets to the point where it produces a little ethylene, then environmentally, developmentally, then you probably want the other fruits to start ripening. So you'll see this effect of ethylene causing ripening. A very important one, and I don't have a picture of this, is that you see a lot of damage in greenhouses. Ethylene is a product of incomplete combustion. So, and I don't know what I did with the slides, but I had these neat pictures of a plume in front of a fan, a heater in a greenhouse, and you could just see the ethylene damage. You could see the closer to the heater, the more damage. And of course, it's a major factor in post-harvest. Now, it's a very simple pathway. And what you should know is that there's basically two parts. The Yang cycle and then the conversion of ACC into ethylene. So here's the part you need to know. The Yang cycle was the conundrum, because ethylene is produced from methionine, and methionine is an amino acid that contains sulfur. Okay? So you smell methionine every time you go by any place where they're doing permanents, because when you're doing a permanent, you are breaking the sulfur-sulfur bonds in methionine, and you're liberating some of the sulfur. That's why permanents smell. But the question was, this doesn't make any sense because we'd be running out of sulfur. So you need to know the Yang cycle is basically to cycle sulfur back, and that the next step is the production of ACC, and then the next step is the production of ACC is the conversion to ethylene. So you're talking about an incredibly short, very rapidly acting pathway. Okay? And you see, we'll talk about today or the next time, there's a lot of work trying to control this ACC oxidase. We want to control it before it gets to the production of ethylene. Again, ethylene is induced by auxin, by, like I said, wounding stresses, and ethylene is produced by ethylene. And almost everything is at the level of this ACC synthase. That's why everyone is so interested in that particular step. Something that's important, and I have this in my lab, and we've used this a lot, is a chemical called Ethylene, which is a fairly stable chemical, and it breaks down 1-to-1 to produce ethylene. So I should have brought the chemical down. There are several different names. I don't think anything is actually called Ethylene, but it's used from everything to maybe thin fruit, change sex expression. I'll try to remember. I can't remember the name of what I've got. Anyway, it's you can get different concentrations of the ethylene depending on what you want to do with it. And there's a new product that we're going to discuss called 1-MCP that we're going to probably end up discussing Wednesday that actually inhibits ethylene production, and that's becoming more important. As problem with MCP is it's a gas too, so it's very, very hard to deal with a gas. Okay, let's talk about climacteric and non-climacteric fruit. When we talked about respiration, we talked about how you have different levels of respiration. And that we get this burst of respiration a lot of times when fruit mature. Ethylene also can change the way a plant grows. And one of the classic things that we see in flooding is something called epinasty. And that has to do with actual irregular cell elongation. And you see this when you see a flooded plant. And the classic is a flooded tomato plant where the leaves are sort of growing down. They're sort of growing like this. And so what you have is ethylene is being produced in the root. That's where the stress is. And it actually causes this irregular elongation of the leaves. And so you can usually tell just by looking if the plant has some kind of a water stress. Now, every time you water a plant, you flood the plant. The ethylene response occurs a little bit later, so you're not going to see this continuous. But if you go, if you go out after we've had a rain for several days, you'll see the plants. They're not really, they're not really wilted. It looks a little bit like wilt, but it's actually their regular growth is causing them to actually sort of look aim downward. Next time you're in a situation where you've got a lot of water in the field or whatever, you'll be able to see this pretty easily to see. Now, ethylene is, the ethylene pathway, most of what we know about ethylene has come from using Arabidopsis. And something that is very classic is known as the triple response, where in the seedling it inhibits elongation, it causes swelling of the radical, or the hypocotyl, and you get an exaggerated curvature. And the reason why these are important is because we can look for mutants, and we'll talk about that Wednesday. So if you have a normal looking seedling here, here's a pea seedling, and you can of course do this Arabidopsis, then you see this if you expose it to ethylene. The beauty of this is, and we'll talk about it again, is that if you want to work with ethylene mutants, then you can expose plants to ethylene and see, instead of getting the classic look, you get that 1 in 1,000 that's normal, which means this plant is not responding to ethylene. So it's a fairly easy way of picking out mutants. Most of the work is what's been done with Arabidopsis. It's been proved in tomatoes because that's the classic, is try to keep tomatoes from ripening. It also induces abscission. And when we talked about auxins, we talked about abscission, and and we went over how abscission actually occurs. It's an interaction between ethylene and auxin. And here you can see plants that have been exposed to high concentrations of ethylene. The one on the left is a normal plant, and the one on the right does not respond to ethylene. So again, it's pretty easy to tell if you have a mutant. Now, actual abscission, and when I'm talking about abscission, I'm talking about the normal loss of leaves from plants. It's a very, very complicated process. And if you think about what's happening in the fall when the leaves are starting to fall, first there's a movement of material from the leaf back into the plant proper, and then after that, then the leaf sort of starts responding to ethylene, so the leaf is basically senescing, which is dying, and you get this very complex process to basically drop the leaf off the plant but seal the point where the leaf is attached to the stem. Now, what's interesting is that in the maintenance phase, which means the leaves do not fall off, auxins keep ethylene low. So as long as the leaf is green, as long as the leaf is producing, the leaf is going to produce a bit of auxin and keeps the leaf attached. So the way I like to view ethylene is like ethylene is just waiting to go. Just there, always there, ready to go. And some other hormone is keeping it from going. Then auxin levels fall, which causes the ethylene levels to rise. And of course, when ethylene rises, it produces more ethylene. And this causes a very specific layer of cells then to break down. Cellulase breaks down the cell walls, then what happens next? Anybody should be able to tell me what's going to happen then. You got the weight of the leaf. What cellulase is going to break down the cell walls. What else happens? Turgor pressure is going to start expanding the cell out. The cell then is, without a cell wall, is just going to blow up. Remember, it's going to have water going in it, and it's just going to expand. And you have then this very specific shedding. So if you look at it, if you look at where leaves actually come off the plant, if you think about it, they all come off at basically the same place, where the petiole attaches to the stem. And if you look at this, you can actually go in and see that it's a very specific layer of cells. So after this, the ethylene starts breaking, causing the production of cellulase, and the cellulase then causes these cells to degrade, then the weight of the leaf is going to cause it to just drop. As the cell walls break down here, the protoplast is going to expand and finally explode. And then this also is sealed, so there's not an opening into the plant. So it's a very complicated process. Now, I mentioned respiration rates, and this is very, very important in fruit and vegetable and any post-harvest production. Anybody had Dr. Cisneros post-harvest class? You go in a lot about ethylene. This is the number one thing that's controlled in a supermarket. Used to be supermarkets had a tank of ethylene in the back, and they'd get their green bananas in, and they would zap them with ethylene so they would start turning yellow. They don't really ripen, they just turn yellow. And that became too much of a risk, and so like now, HEB zaps their bananas in San Antonio before they're distributed. And so what a supermarket is trying to do, or anyone is trying to do when we're looking at post-harvest, is control ethylene and control respiration, because obviously the faster the respiration, the quicker the material is going to break down. So when you go into the supermarket, things are in the cold because they're really trying to keep the respiration rates down. You also go to the supermarket now and you see more and more products that are ready to eat. You pay some astronomical amount, like a equivalent of 2 apples or $3.99 or something, where they're cut into slices. Probably more research is going into the kind of plastics that you put those products in. They're not just saran wrap. They're very complicated, sometimes more than one level of barrier that's going to allow CO2 out or O2 in at a different rate or absorb ethylene. A lot of work goes into controlling the respiration rates in fruits and vegetables. So if we look at some things that are have different respiration rates, asparagus, if you go and look at asparagus, it's usually stuck down into some ice, because you're trying to lower the respiration rates. Sweet corn, if you really want good sweet corn, the joke is you have to take and boil the water in the field and drop the sweet corn in the water in the field. Actually, with sweet corn, they do something called hydrocooling. So you'll have an 18-wheeler, where the back of the 18-wheeler is divided up into sections of very, very cold water. And as soon as the corn is harvested, it's dumped into this water, this very cold water, to lower the respiration rate, even before it goes to the packing shed. So it depends on where the best way to store these things are basically just done over time. Here's the classic response that we see, okay? We start seeing CO2 production, and then at some point we get this burst of ethylene. And this burst of ethylene causes CO2 production to go crazy. This is ripening. Now, again, fruits are not designed for us to store. They're designed to ripen and disperse the seeds. So a lot of work goes into disrupting this part right here. Okay? Mostly the classic, of course, is in tomatoes. There are fruits that are not climateric, which are over here, and this is called the climateric response. And there are fruits over here that do have this response, okay? Tomatoes, of course, is classic. Apples, in Washington State, they have storing apples down to a exact science. There's a USDA center in the center of the state. And apple producers send their apples in on a regular basis, and they will tell them exactly down to the day when to harvest the apples. And it's all about controlling ethylene. They have their warehouses that are as big as this building, where they store apples in controlled environments. And they're neat because they're sealed, but they're so big, changes in the barometric pressure would cause the thing to produce leaks. So they have these huge bladders that look like an accordion. They're just these rubber bladders that you can walk in. They're so big. And so as the air pressure changes inside and out, those bladders will expand and contract. They've actually got it down to where they can store apples now for over a year, which may not be that great for their business. Other things, mostly apple. I did a postdoc in Washington State. That's what I know about apples. I know about tomatoes because I did my PhD in Florida, and at that time all tomatoes came from South Florida. Tomatoes are picked green, put into a truck, and they know where the truck is going, so they know when to zap it with ethylene, so that when that truck arrives, so if it's going from Florida to New York, they know when to hit it with ethylene so that when the truck arrives, the tomatoes are just turning red. Now, in the way we control it, there are some simple things. There's silver nitrate. Silver nitrate is still used, but not as much because silver costs some money, and there's a problem of polluting the environment. But silver nitrate's used a lot still with fresh flowers. CO2, we can use CO2 in the supermarket. If you buy corn. If you buy corn, where you buy the corn that's in the little tray that's coated with the plastic, that's designed to keep a certain CO2 level inside the plastic and an O2 level down, so you can control the respiration rate. So that's why you're paying so much more for that corn. They're extending the shelf life of that sweet corn. Things that are used commercially are these two, ABG, AOA. Ethylene we talked about. And a new one that's on the market now is MCP, that's used commercially, and I think I'll talk about AgroFresh a little bit later. AgroFresh has developed a powder that is basically a polystyrene ball that has MCP trapped in the powder. That way you can make up a solution and spray it. And so they've been able to successfully treat apple trees. Otherwise, the only way you can use MCP, and it's used, is if you have a high-value plant, you can put it in an airtight environment, a growth chamber, and add MCP to the growth chamber. Okay. Here's something you're going to see wherever tomatoes are processed. This thing is an ethylene generator. And the thing is that tomatoes are not picked green. They're picked at a certain stage that's not quite green, and then the ethylene is added later. What's a vine-ripened tomato? What does it mean if a tomato is vine-ripened? No, you see that. You see that in the store. Vine-ripened tomatoes are the 3 or 4 tomatoes that are still attached to the vine. But you can also see vine-ripened tomatoes that aren't attached to the vine. What makes a tomato vine-ripened versus non-vine-ripened? It's almost pink when it's picked. It's not red. It has an actual little bit of pink color. That's, quote, legally a vine-ripened tomato. Just like a fresh turkey, can only be frozen like to 28 degrees or something like that. So a fresh turkey can be frozen. It's just not been blast frozen at minus 40 or whatever. So remember, these are legal definitions. Those vine-ripened tomatoes that you see, those are actually first introduced by, I want to say the Dutch, who sold them for an ungodly amount of money because they were literally actually ripened. Most vine-ripened tomatoes are still picked way before they're ripe. And the big problem is, if you take a green tomato and expose it to ethylene, it turns red. It doesn't ripen. It'll get soft, but there's no flavor development that you get. The thing with the tomato that you leave in the field until you eat it is that you have all the organic acids and sugars going into that tomato, and you're getting a normal ripening process. Once you pick a green tomato and zap it with ethylene, you get partial ripening. You don't develop the acids. You don't develop the flavor. Okay? Here's an old picture of some plants that were treated with silver nitrate. So 14 days later, you get a really nice, you still get a nice color. Here's the official color classification. Anybody ever use one of these? Somebody? If you go to a packing shed, you'll see these, and this tells you what you're looking at. So here's breakers. They're usually picked at a breaker stage. If you pick a tomato too early, it won't respond to ethylene. So you got to pick it at the breaker or turning so that when you zap it with ethylene, it'll actually turn red. So you can't pick a real green tomato and zap it with ethylene. Then you get to the pink and see the turning. If you have a really good imagination, you can say, well, yeah, that's some red there. And then, so they're either picked at the breaker stage, and vine ripen's going to be here, but here or here. Okay? And let me stop there, and I'll pick this up later. And any questions? Anyone? Yeah, this sort of makes going to the supermarket, sort of takes the mystery out of it, but it's very important.